Welcome everybody to the February 24th, 2022 meeting of our working group. We have two great talks lined up, and we very much appreciate everybody coming today. I need to remind you that the meeting, as always, is live streamed, recorded, and publicly available. So please do keep that in mind in your participation. As always, Reinhardt and myself are here to take suggestions, comments, criticisms. Jim Sluka is one of the organizers as well. He's available to help out. And Lorenzo has been very generously working on organization and also the scientific content of this working group. So we are all there to help out. Please help us distribute our information. We'd like to have more people participate. We are always eager for your help in getting the word out. In particular, we have our YouTube archive of recordings, more than 100 talks now. Please help us distribute those and make sure people know about this great resource. We have the Twitter feed. It's better if people use it. So please help us populate our Twitter feed and publicize what we do. Are there any announcements for today before we get started with the scientific program? Reinhardt, I guess we have a meeting of the digital twin working group tomorrow. Is that right?
That's correct. If you're signed up, please join us. If you're not signed up, please join us. If you need a link, let us know.
And I guess I should say that I am again looking for a computational biology postdoc. So if anyone has somebody finishing up who would like to come to Indiana to work mainly in developmental biology and cell signaling, I would be delighted for you to send them my way. Any other announcements before we go on? No. So our schedule coming up is that we have single speakers booked for all of March. We have Alex Hoffman, Benjamin Tanover, Sally Sieberts, and Susan Schreiner speaking. We have opportunities if you would like to contribute a second talk for any of these meetings. And we are always looking for your suggestions going forward. We particularly welcome suggestions for people who might not be the obvious suspects: clinicians, experimentalists, drug developers, people doing modeling at other scales from molecular to the whole world. Anything that you think you would like to hear, people doing interesting work. Lorenzo is extremely good at convincing people to come and speak to us. So please do encourage him and us to invite people you would like to hear from. And also feel free to volunteer yourself, your postdocs, your graduate students, because you don't have to be a senior scientist to speak in our meeting. So during the meeting presentations, please mute your microphones. We'll follow our standard protocol, which is we will have both talks and then a period of open discussion up till we either run into 4:30 or our speakers need to leave or we run out of questions. Our first talk is from Hayriye Gulbudak from University of Louisiana, modeling across scales feedback of infectious diseases. As people know, I don't do elaborate introductions. I've already taken enough of our speaker's precious time, and so I will turn it over to you.
So Dr. Glazier, you want me to share the screen, right? Yes, you. Here we go. I think we're all good. Just let me know if the slides are not moving. It sometimes happens. So I want to check with you. Can you see that slides are moving? Perfect. Okay. Hello, everyone. Thank you very much for your interest in my talk. And thank you for the invitation. I'm really honored to be here and share my recent research with you. Today, I will be talking about modeling across scale feedbacks in pathogen variants, host immunity, and disease control. I'm going to in particularly focus on dengue, one of the recent work I did, and mention some of the future work. I think given that the time I have very short, so I just didn't want to, give a full scale talk about that. But I will probably on and off talk about stuff with not dengue as well too, and application of the small scale models. So epidemiological and immunological models have been traditionally treated separately, and however, the between host and within host scales of infection are linked and together determine the dynamics and evolution of the disease. There are several motivations for models connecting scales, for example, pathogen host evolution, small-scale data fitting, sensitivity analysis, incorporating within vector, mosquito, viral kinetics, and tracking immune status of individuals in the population. So today, the focus of my talk is formulating a model which allows you to track the immune status of the individuals in the population, while can still keeping track of the transmission happening on in the population scale. So this is a very simple, the schematic diagram for a vector-borne disease. You have susceptible vector population at time t, total number of infected vector population at time t, and susceptible host population at time t, whom have pre-existent antibodies Y, and infected host population, density of infected host population infected with strain K at time t, whom initially had the pre-existing antibody level Y not, but currently have antibody level Y. And we have also individuals recovered from this strain K, but currently have the antibody level Y. So the type of model I will consider, as I mentioned, is going to keeping track of individuals with their immune status. However, to do that, you would like to link your population scale epidemic data to within host scale viral immune kinetics. So in the within host scale, the model successfully incorporates heterogeneity among the infectiousness of the individuals in this way. So within infected individuals, we know that there's an interaction going on between pathogen and two types of immune response. In this case, we have IgG cross-reactive antibodies and IgG specific antibodies. Soon I'm going to mention you what these two immune response does if you do not have an idea about that. So this is a simple schematic representation of dengue I will mention. So assume that when the mosquitoes bite the host, it releases some amount of pathogen, and then there are two types of immune response. So we have specific IgG immune response, and we have the green ones, basically the cross-reactive non-specific IgG immune response. So the more specific IgG immune response you have, the more they're capable to kill the virus. However, for the non-specific IgG immune response, it takes large amount to kill the virus. So one of the interesting things about the dengue is that, unfortunately, although it's shown that for dengue, we have 4 different strains, strain 1, 2, 3, 4. And it's shown that once you recover from the first strain through, given your primary infection, actually, later on, once you recover, you never, pretty much you have lifelong immunity. However, when you get infected with the second strain, it's possible that due to pre-existence antibodies you have, not only the prior pre-existing antibodies do not protect you, it does not give you partial protection, indeed, you might get more severely infected, means that you might experience dengue high fever or dengue fever. So this phenomena is known to be as antibody-dependent viral growth enhancement, and the mechanisms behind of this phenomena is just yet to be understood. So we basically wanted to build a model where we can keeping track individuals with respect to the amount of the pathogen and specific and IgG and non-specific IgG antibody level. So when we do so, we can actually once the individuals, among the recovered individuals, we can say something whom going to experience the dengue high fever. So one of the story about this work is that actually me and my collaborator, when we were reading science article about dengue, we it was very interesting. An immediate question was that someone who was very interested in multi-scale modeling was how we can actually formulate the dengue in this multi-scale modeling framework. So it turned out that, so for example, one of the story in Philippines some years ago, they applied large-scale vaccination. And then when the dengue outbreak happened, actually they observed fatality among the children. And what the government did is that government basically, if I remember the story correctly, threw the scientists in the prison, blaming that it was their fault. So this is the story. The thing is that because of this AD, antibody-dependent viral growth enhancement, so there is the assumption that there is a risk window of pre-existence antibodies. The individuals who recovered from the prior infection, if their antibody level in that window, when they infected with another strain, they will experience the dengue high fever. If they either recover from dengue high fever, get lifelong immunity, or they die. If they don't die, then they get lifelong immunity. So our first question was that whether pre-existing antibody levels can be the determinant of the whole infection severity. So to do so, we basically built a coupled OD system where we have X represent the viral load, and we have Y represents the cross-reactive antibody, IgG antibody load, and Z represents the specific IgG antibody level. Because the IADE is, the individuals who experience dengue high fever and dengue fever is mainly experienced this thing due to their pre-existing antibody levels, which is cross-reactive IgG antibody level. And soon we are going to build, and I'm going to mention how we formulate the population scale model, which keeping track individuals with respect to the cross-reactive IgG antibody level they have. So on the Wittenau scale, we assume that so there's a competition between these two types of immune response, specific and non-specific IgG immune response. And we basically put the competition between two immune response through this functional form. And we also assume that, so if you can see that there is like here, we have two minus terms. One of them is just, cross-reactive IgG immune response and specific IgG immune response, both are capable to kill the virus. So when you see this Z term, it's a negative term, it always kills. The more you have specific IgG response, the more it kills. However, when it comes to the amount of the cross-reactive IgG immune response, it only takes good amount of having good amount of cross-reactive IgG immune response to kill the virus. Otherwise, so this is a saturating form, and we have a heal function form. So indeed, you will see that when the cross-reactive IgG immune response is low, this term become negative, so it's kill the virus. When it's larger, it's kill the virus. However, when there's an intermediate level of IgG, cross-reactive IgG immune response, where this term becomes actually positive. So the first thing we tested basically whether this model can mimic the characteristic of the secondary infection. So when you look at the graph, on the x-axis we have time since infection, and on y-axis we have viral load, which is the red curve, and we have the blue curve basically shows the cross-reactive IgG immune response. We didn't basically plot the specific one because the ones we are interested in is the cross-reactive one, the ones actually cause the AD. So as you can see, when they assume that individuals get infected, once the virus start to grow within the individuals, then the immune system, it triggers the immune response, and with a lag, and then it's killed the virus, and eventually these individuals recovered, and now they get amount of antibody level. So assume that there is some years passed, some time, assume that 2 years, and then there's a waning going on. And then by the time this individual experience secondary infection with different strain, so it's possible that if the individuals have larger peak viral load and then a shorter time to peak viremia and higher maximum clearance rate, then we say that this individual experiences the dengue high fever or dengue fever. So we see that actually our model produced signatures of the severe dengue infection. So when you look at this figure, on the x-axis, we have pre-existing antibody level Y naught, and the y-axis shows the peak viral load. So you can see that actually there's ideally you think that the more you have antibody response, the more you should have protection, right? It turned out that this actually may not be true. So there's an intermediate value of this antibody level which actually the peak viral load is larger. So we basically observed that host infection severity, peak viral load has a unimodal function of pre-existing antibody level. So some of all the characters, so basically with the secondary infection, we see that our model produces all the characteristic of the secondary infection, which is causing the dengue high fever. So, we mathematically want to see that whether this is the model which represents the characteristic of dengue high fever. It turned out that yes, and we actually see that given certain parameters, we see this unimodal shape, means that the severity of infection is actually unimodal function of the pre-existence antibodies. So it's basically saying that if you remove any of the immune response, IgG or cross-reactive or specific immune response, you will not see this interesting characteristic of the secondary infection. So I want to say the second question we want to, linking the epidemiological scale disease transmission, we wonder whether within host pre-existing antibody level can be the predictor of disease outcomes on population scale. This is something really important for COVID too, because with the COVID, we know that ongoing issue, we get boosted, we get vaccinated, but in the meantime, we are losing our immunity, we are susceptible against certain level. And then there's all this conversation of like things, the herd immunity constantly moving, and we don't know whether, so, it's just, things are wild. Some people just getting vaccinated, they boosted, some people losing their immunity. So there's constantly shift and they become more susceptible. In the meantime, again, the disease incidence dynamically is also changing. So on the Vittinus scale, as you remember, I described this characteristic of, the model structure and how the solutions mimics the dengue high fever with AD. So on the population scale, we want to formulate the model in a way that when susceptible individuals with certain pre-existing antibodies, when they experience the primary infection, let's say with strain K, their antibody level move to here. So this x-axis basically shows the pre-existing antibody level. And through time, we said that, okay, this individual, once they recovered, then when as time passed, they lose their immunity. There's a waning happening. So there's just waning. And the cross-reactive IgG immune response decreasing, pre-existing immune response. By the time these individuals is infected, if their pre-existing antibody level, which is basically this is what I'm talking about, in this risk window, we know that these individuals will be the one experiencing the dengue high fever. So assuming that these individuals, they either die, or if they don't die, they get permanent, cross immunity, and then So you want to just formulate a dynamic model in a way that not only keeping track the individuals with their immune status, also just keeping track of all this waning and then death process and a birth process, along with like reinfection with another strain, etc., etc. So what we did is that we come up with this to track the immune status of the individuals on the population. We formulate an antibody size structure, PD model, which keeps track the individual according to their infection with strain K, primary infection. So they get infected and then they recovered. So they move to the recovered compartment. And these individuals, recovered ones, can also experience the secondary infection. But again, if their antibody level in this risk window, these are the ones going to experience the dengue high fever. And then in the meantime, individuals lose their immunity with waning. And then we have also to link the within host scale by, we formulate these linking functions like the transmission from infected host to susceptible vector, and along with the recovery rate and waning, we choose as functions of the within host viral immune kinetics. Because the ideal is that the more individuals have virus, the more likely they are going to cause more infection. And then to be able to say in individuals completely recovered, you need to check to see whether their viral load is decay and they have their antibody, and you need to check their antibody level, whether they saturate or not, to say that they recovered individuals. So to do that, to link the immunological and epidemiological scale, we basically, first of all, as I mentioned, that we formulate the host vector transmission as the sigmoid function of the viral load within the host. So based on this formulation, based on this work, which is published by Handel and Rohani paper in 2015, they show that, for example, on the x-axis, when they have like viral load, and on the y-axis, they show that proportion of the successfully infected mosquitoes. So they see that basically what they are saying is that host to vector transmission is a sigmoid function of the viral load, the pathogen load. So there are different type of examples of recovery rates with basically formulation we consider in our simulations, and soon I will be talking about them further and why they're important. So by considering to be able to do this numerical simulation, so it's a very complex model. Usually, some of you might be familiar with the age since infection structure model. So you have two variable, actual time t, chronicle age, time, and you have infection age, means that how many days ago individuals get infected. But when you do with the numerical part of this work, usually it's easier in a sense that you have like actual time t and time since infection go hands by hands, and there's a linear relationship. But in our model, we have two variables, and one of them in the actual time t, and the other, and we are keeping track of individual not with their time since infection, with their non-specific IgG antibody level. So we don't have this nice linear relationship to be able to handle the simulations. So, but we had to get around it. It's been a very challenging problem. But you want to you would like to do error analysis to be able to see that how your numerical simulation is doing well, your algorithm. To do that, having some analytical work on the hand helps a lot, because, for example, we can derive the equilibriums where they are the special solutions, and we can just basically simulate the solutions and see that whether eventually they are going to converge. And this is going to kind of help us to do some sort of error analysis. So assuming that under simplified condition, assuming that we have this waning rate psi and piecewise constant function, which we are assuming that there's a temporary cross immunity, and assuming that that rate is greater than waning rate, and then considering the susceptible antibody distribution as like transient normal, actually we can define, given that how many individuals are, how much antibodies recovered individuals have, we can determine among these recovered individuals whom going to experience the dengue high fever when they infected with the secondary infection. So for example, on this figure, on the x-axis, so we considered here death larger than the waning rate. So what we did is that we simulated recovered individuals with antibody level Y with respect to their antibody level, and then later we basically plot the secondary infected cases, and this figure basically shows that the recovered individuals here in this whom have antibody level between, let's say, 0.17 and 0.85 is the one going to experience a dengue high fever. So also assuming that in other case, on the other hand, if we assume that individuals, the waning rate is larger than the death rate, we can see what recovered individuals with what antibody level going to experience the dengue high fever. But you can see that the recovered densities goes to infinity under this case, which we know, because of the assumption that waning rate is larger than death rate, and we know that when you integrate it, it's going to be finite. So what we did is that basically to attack the numerical part of this work, we consider a first order finite difference scheme computing solutions to the antibody level structure PD vector host model with the stored within host calculations. So remember, we linked the epidemiological scale model in a way that the transmission rate, recovery rate, waning rate, they're all they are the function of within host coupled viral immune kinetics components. So for example, two components of the finite difference scheme is like we have the infected individuals with strain 1, recovered individuals with strain 1, where IJN denote the index for discretized pre-existence antibody level, current antibody level, and time respectively, and M is the maximal antibody level. So the last question, which is the current world, what I'm trying to see with my students, my graduate students, Caleb McDonald, who's going to graduate in May, soon to be postdoc at Tel Aviv University, we actually started this project this semester. It wasn't a part of his thesis, but he's so good with data that I wanted to make sure that, I wanted to work with him. So he was very excited. So one of the questions we are targeting is that we want to know on the population scale, how can vaccination be employed to eradicate the disease while avoiding possible severe infection in vaccinated individuals, all at a feasible economical cost. So I want to show you this figure. So for example, the blue figure is the reproduction number, given that means susceptible pre-existent antibody level, Y. And the red figure is basically just shows the individuals who experience the dengue high fever. So as you can see, what is good for the population scale may not be what's good for the individuals. So the orange curve is about how many individuals experience the dengue high fever. Red, blue curve is about the population scale, whether how much the disease is controlled given. So the idea is that an ideal vaccination program should be in a way that not only control the disease, but also protect the individual from the dengue high fever as well. So what we are trying to do currently right now, we are trying to fit the multi-scale data within host viral immune data in the immunological scale, and we want also to fit the population scale disease incidence data just to inform the public health agency, maybe just to say whom to vaccine and then when you vaccine the recovered individuals, what level on pre-existent antibody level the antibody should they have, and vice versa. And then the idea is that in terms of, this is not necessarily a predictive model we use for prediction, but we want to try to understand things qualitatively that we can actually get some helpful insight and help, inform the public health agencies. So I'm just going to stop here. And these are some of the relevant, literature and recent work. I'm working with my graduate student, Caleb McDonald, Maya Marcheva, Nejibe. I have been working through the years this work. Vincent Canatora, Cameron Brown, Zulin. She's currently, she's Max postdoc, but she was Max postdoc, but currently an assistant professor at University of Texas and San Antonio, and Fabio Milner from Arizona State University. Sorry about my nervousness, and then ignore the baby crying at her. Somebody's taking care of her. So this is it, and thank you very much. And I would like to, acknowledge the funding agencies for their support. And I got funded through when I was a grad student. Actually seeded, it wasn't part of my thesis, but it seeded the idea of my current ongoing work when I was there. And NSF, also Simon Foundations for their support. And through that way, I able to fund my students, and then this works happen, be able to hit this works out there. Yeah. Thank you very much.
Thank you so much. We really appreciate the illustration of coupling in-host and population scales, which we talk about a lot, but don't see done so often. So we will go on. Ruchira, I think the request was that we hold questions until after the both talks, if that's okay. So we will continue with Abba Gumel from Arizona State, who's going to be talking about mathematics and the renewed quest for malarial eradication.
Thank you so much for the organizers for the invitation to speak. And great to see my good friend Hayriye Gulbudak giving an excellent presentation. She always does a very good job. And great to hear the baby also crying. So maybe the baby can present the talk on my behalf. So, no, but excellent. So I'm going to talk about malaria, the renewed quest for eradication. But before starting, I need to acknowledge also the sponsors. MSRI has been very supportive of some of our training activities, our joint program, especially that they hosted last 2 years ago, the NSF Simons Foundations, and I'm affiliated with the University of Pretoria in South Africa. So through that, I have funding from the National Research Foundation of South Africa as well. Okay, so this talk fits in nicely from Hayriye's talk, because she talked about dengue and other vector-borne diseases, but so vector-borne diseases in general constitute about 17% of all infectious diseases of humans, killing about 700,000 people annually globally. So of the vector-borne diseases, malaria is the most important, causing, actually before 2000, it used to kill 1 to 2 million people each year globally, and with 70% or more of those deaths occurring in children under the age of 5. So we lost about 150 to 300 million in the 20th century alone. Dengue, as discussed by Hayriye a moment ago, it's a very serious infectious mosquito-borne disease. with 4.4 billion people actually in over 129 countries at risk of acquiring dengue infection. And there are many other vector-borne diseases, as some we are familiar with, like chikungunya, Zika, and Rift Valley fever, yellow fever, and so on. West Nile virus is coming back again here in the state of Arizona last year. Many of these diseases are zoonoses, and this lays the foundation of most of what I'm actually trying to do nowadays. That these are diseases that emerge or re-emerge due to what we as humans do to the environment. So it's the interaction between pathogens, host, and the environment that give rise to many of these deadly diseases. So this is the One Health initiative that we've been talking about. You know, in order for us really to understand what's going on, you know, how to come up with best or good strategies, excellent strategies for controlling the spread of diseases, we really need to understand what's going on in the wild as well. So the interaction between humans, between pathogens and the environment is very critical. So there are climatic factors and non-climatic factors that are at play here, but I'll talk about this later. Anyway, so of all the vector-borne diseases, malaria is the most important, largely because of the burden that it inflicts globally, and particularly in sub-Saharan Africa, and in some parts of Southeast Asia, some parts of Latin America. So, okay, some basic information about malaria. It's transmitted between humans through the bite of an infected Anopheles female mosquito. So the female mosquito takes blood milk from humans for the protein in our blood needed for the process of development and maturation of eggs. Okay, so that's the reason only female mosquitoes bite humans, male mosquitoes do not bite us. So this is, and the parasite actually that causes Plasmodium, malaria is Plasmodium, okay? And there are many, the mosquito vector is Anopheles, so about 450 different species. Only about 41 are major vectors that can transmit to humans. And to be a major vector, you have to have three properties, basically. First, the mosquito has to habitually bite humans. And second, it has to harbor Plasmodium to be able to carry the parasite. And third, it has to live long enough to complete this progonic cycle, which I'll talk about later, which is basically the process of the time period between the initial infection of the mosquito and when the parasite matures in the mosquito so that it can be transmitted to the next human. There are 5 different species of the parasite, and the usual symptoms for malaria are listed here. But of the Anopheles mosquitoes, Gambia is the most important one, is because it's the most prevalent in sub-Saharan Africa. It has very high vectorial capacity, and it thrives in conditions that are associated with anthropogenic alteration of the environment. So man-made alteration of the environment, such as deforestation, construction, agriculture, human settlement, livestock, and so on. And COVID made malaria much worse. And if you compare the data for 2019, here, this is number of people who died globally of malaria compared to 2020 is much higher, and case data as well. So of course it's expected, and that's what happened. And this is a typical life cycle of the Anopheles mosquito. So think of this as a female Anopheles mosquito. The first order of business is look for a human host to bite. If it succeeds, it goes into, this is the first stage of the gonotropic cycle. Gonotropic cycle is the host seeking cycle, right, of the mosquito. So the first stage is to look for a human host to bite, take blood milk from. And if it succeeds, then it rests on the wall. It goes to second stage of the gonotropic cycle, resting on the wall for the process of maturation and development of eggs. If that is completed, it goes to stage 3, which is to find a breeding habitat to lay the eggs. And then the eggs hatch into larvae. They're instar stages, based on the size. They live inside the water, and they compete for resources and space. And this larvae become pupae, and then it leads to an adult mosquito, and the cycle continues. So this is the basic life cycle of the malaria mosquito, Anopheles mosquito. And the first control strategy against malaria was actually not formulated by mathematicians, but by a medical doctor, actually going back to Sarona Rose, who won the Nobel Prize in Medicine for actually determining or for finding that actually in the midgut of the mosquito, they found Plasmodium, and that plasma. So that was the first time actually to determine that malaria was caused by mosquitoes. Mosquitoes transmit malaria to humans. So Sarona Rose did that and won the Nobel Prize in Medicine in 1902. But this is the basic model that he used, okay? So X is the population of infected individuals, infected humans, and Z or Z is the population of infected mosquitoes. So total human population is H and total mosquito population is M. So H minus X is this susceptible population of humans, right? So if a susceptible human is bitten by an infected mosquito, this is the proportion Z of infectious mosquito, and this is the bite rate A, and this is the probability that this infection leads so this bite leads to infection, and M is the host vector ratio. I mean, so this gives you the total number of new cases generated by one infectious mosquito, and R is the recovery rate of humans. And similar equation here for infected mosquitoes. So M minus Z is actually the susceptible mosquitoes, and they bite infected human, this proportion X, and the probability of infection C, and A is again the host vector, the bite rate, sorry. And mu is the average lifespan of the mosquito. So a very simple model, and calculated the reproduction number, which is the average number of new cases generated by a typical infected individual or infected mosquito, introduced in a completely susceptible mosquito or human population. And it's given by this, all these parameters of the model, okay? So M is in blue because that's what Sarona Rose focused the intervention on. So if R naught is less than 1, you can control the disease. If it's greater than 1, the disease persists in the population. So clearly you want to minimize the numerator. So M is in the numerator, is the host vector ratio. So what Sarona Rose told the WHO at the time was, well, reduce the host vector ratio, reduce the number of mosquitoes per person per unit time, which makes sense because it's in the numerator. So minimize M, you decrease R naught. And that led to the foundation. Okay, that was the very first scientific control effort against malaria, basically. But his approach was to focus on destroying mosquito habitats. So more like larviciding, just using habitat destruction as a strategy. But this laid also the foundation for threshold theory. Okay, so we do not have to kill all mosquitoes to effectively control malaria in Western Europe. What we needed to do, what Sarona Rose said, was bring the mosquito population below a certain threshold, and now you have a chance to control it. So, and that led really to the notion of R naught, right? R naught less than 1, you have a chance to control. Greater than 1, the disease typically persists in the population. So DDT was invented or synthesized in 1939 and was widely used during the Second World War, and also used widely in agriculture. But then George McDonald came along in the 1950s and extended the Sarona Rose's model. Notice here that once a mosquito is infected, it's instantaneously infectious in this equation. Okay, so, but George McDonald added the time delay between the initial infection of the mosquito to when the mosquito becomes infectious. And that's to capture this progonic cycle. And through that, by adding the time delay, the control effort now, the control strategy was changed completely from Sarona Rose's idea of habitat destruction to focusing more on adult mosquitoes. And that's what is happening up to today. The focus on malaria control is more on adult mosquitoes through the use of indoor residual spraying or bed nets and so on, not necessarily larviciding. Okay, so the early control for malaria, so the WHO, because motivated by McDonald's work, decided to embark on this malaria eradication program in the 1950s, which unfortunately was disbanded in 1969 because of widespread resistance to DDT, which is again the focus of my lecture today. And so anyway, this was disbanded, but then in the year 2000, 2015, there's wide-scale use of insecticide-based intervention, which largely focus on use of bed nets. So a human sleep under the bed net. The bed net has two main properties. It either kills the mosquito on contact or it repels it away from getting into the net to have access to the human. And there are 5 classes of ingredients that are used in this indoor residual spraying and also the long-lasting insecticidal nets. All 5 are used in IRS, but only one is used in this net, which is the pyrethroid. The pyrethroid has 2 main properties. It has this low mammalian toxicity, so it's not very toxic to the human, but it also has this repellent property that it repels mosquitoes from even getting into the home of the person sleeping under the net. Unfortunately, the mosquito has selected also for resistance against all 5 currently available chemicals. So that lays again the foundation of what I want to talk about. There's now concerted global effort to eradicate malaria because of this dramatic reduction recorded during 2000-2015. So the question is, can we still eradicate malaria and using existing resources, existing insecticide-based intervention? The main mechanism is still the use of insecticides. So can we eradicate malaria using those interventions? Okay, so unfortunately, there's 4 main stumbling blocks towards malaria eradication. One is, as I mentioned, resistance to all insecticides, all currently available insecticides used in these 2 controls, the best that we currently have. And second is anthropogenic climate change. Climate change affects all aspects of the life cycle of both the vector itself, Anopolis, and the parasite, Plasmodium. Okay, so with respect to for example, in the vector, with respect to feeding, with respect to fecundity, with respect to host seeking, and all of those things. So maturation, survival, all of those things are affected by changes in, for example, temperature. And the third one is parasite resistance to the best therapy that we have, which is artemisinin-based therapy to treat people with malaria. So that's evidence now for that as well. That's also the problem of migration, mobility, rural, urban, and changes in land use. But I'll focus on the first 2 for this talk. That's currently no safe, effective vaccine. 2 very promising ones are in the pipeline. I expect probably by end of this year, we may get FDA approval for a really good vaccine against malaria, but we're not there yet, but we will be hopefully soon. Anyway, so my objective is, okay, can we eradicate using modeling? Okay, can we eradicate malaria using existing resources? So I'm going to focus only on the impact of climate change and insecticide resistance on malaria transmission and control. And to do this, we need to have some kind of genetic epidemiological and methodology modeling framework to achieve this. Okay, but before going into that, I want to make a case that malaria is as complex as they get. So first of all, there are how many cycles? 4 main cycles associated with malaria. The immature cycle, right? Eggs, larvae, pupae, adult mosquito. That's complicated itself. Second is the gonotropic cycle, the host seeking cycle that I mentioned before, 3 stages. Stage 1, to look for a host. Second stage is to find a place to rest for the process of maturation of eggs. And the third is to find a breeding habitat to lay the eggs. And the last 2 are what the parasite does in the human. So it's a schizogonic cycle, the process of the maturation of the parasite in the human and also in the mosquito. Okay, that's the progonic. That's one of the smartness or how very smart malaria is. Malaria mosquito is the parasite is. It survives in 2 hosts, in humans and also in mosquitoes. And there are multiple parasites. I mentioned there are 5 of them. Right now, there are multiple hosts. It survives in the mosquito, in the human, and also non-human primates, and multiple vectors, about 41 that can give us malaria. So at the very least, this is a multi-host, multi-vector, multi-parasite kind of system, complex system that we have to deal with. But there's also a problem of timescale. Okay, so parasite timescale is in the host. Okay, it's in the scale of like a few hours or days. The time scale in the vector, the malaria vector time scale, so they typically live to be like 21 days. And the human demographic time scale, which is like in the United States, is like 80 years. So multiple time scale problem. There's also fitness cost of resistance, right? Okay, so incorporating relevant data, resistance to insecticide. Okay, how do we quantify all of this? Anyway, so this is as complex as it gets. And this requires really, really extensive modeling to really understand what is going on realistically. So high dimensionality, high nonlinearity, serious mathematical, statistical, and computational challenges. Anyway, so the first model I want to talk about try to address all this. So the long-lasting insecticidalness and climate change. So we have the aquatic stages represented by eggs, larvae, pupae. Adult stages captures the 3 gonotropic cycles. So SEI, susceptible, exposed, infectious for each of the gonotropic cycle. So X is stage 1 of the gonotropic cycle. Subscript Y is stage 2. Subscript Z is stage 3 of the gonotropic cycle. The reason we have to do it this way, because as Einstein says, things should be made simple but not simpler. So Sarano-Rossi's model has only 2 equations, but to really understand what is going on, you need to capture all these main cycles associated with malaria, which at the very least, we have to have some kind of these complexities. And this leads to a model with like 23 differential equations. And I'll just show quickly what happens. So if you are, this is a susceptible mosquito in stage 1 of the gonotropic cycle, the first order of business is to seek for a host, and this is happening at rate BH. And this host can be sleeping under a bed net with probability pi h, or not sleeping under a bed net with probability pi u. So this is the probability that the host actually finds a host that's sleeping under a bed net. So when that happens, this mosquito can die, can be killed by the net with this probability, epsilon di p, or it can get deterred and it comes back and try again. And so if it succeeds in biting this host. And if the host is infected, then the mosquito goes into the second stage and is the Y stage, but infected, and then it can go to the third stage to lay the eggs and so on. So it's a simple strategy which captures both the gonotrophic cycle, the sporogonic cycle, and the aquatic cycle of the mosquito. And all of these things are dependent on temperature, at least water temperature and air temperature. Okay, so a simple model, which I'll probably skip because of time. The only thing to highlight is the mosquitoes that are laying eggs are those in the third stage of the gonotrophic cycle, the Z stage. Okay, they lay eggs at this rate and it's modulated by this logistic proliferation, just to make sure the number of mosquitoes, number of eggs laid are not infinite. So we just put a cap on them. And then TW is water temperature and TA is air temperature and so on. The functional forms of all these temperature dependent parameters are estimated from data. Okay, so we have colleagues who do the experiment in the lab to subject the mosquito to different temperatures, and we use the data to estimate those parameters. And so the usual R naught, we calculated that, and I think I'll probably skip the theoretical part if somebody wants to have a question. This system has a backward verification. What it means really is reducing R naught less than 1 is necessary, but it's no longer sufficient. We need to do a lot more. So the presence of backward verification in this model and the transmission dynamics of malaria makes its control more difficult, because we have to really reduce R naught much, much lower than 1, not just enough to keep it. So we do not have this coexistence of two stable attractors, but I can answer questions if they are related to this. Anyway, so the first question, okay, so is there optimal temperature for maximum malaria intensity? And so we simulated the model to show that, yes. So we see that the R naught increases as the temperature increases from 15 degrees to about 30 degrees, we reach a peak here. So this is when we have maximum malaria intensity, okay? So that means this is the best time to start intensifying control effort. That's an optimal temperature within which you see a lot more mosquitoes, a lot more malaria. So you intensify, and this is, we have similar plot here with respect to this inoculation rate, EIR. With respect to also protected humans, that's in blue, and unprotected humans, those are the ones that do not sleep under abandonment. So you see the EIR, the extrinsic inoculation rate is much higher if you have a higher proportion of individuals in the community that are not sleeping under a bed net. And this is for the overall, so you add the two together. The blue are the ones that are sleeping under bed net. Anyway, we have been able to find optimal temperature within which we expect a lot more infections, a lot more malaria, a lot more mosquitoes, and that's when we should focus our control effort. And we try to also do a GIS mapping to see what is going on subject to different global warming scenarios. So let's see, okay. So first of all, we showed, okay, we had this data correlating parasite rates in kids between the ages of 2 to 10 in the year 2000, before this massive wide-scale use of long-lasting insecticidal nets I mentioned before, from 2000 to 2015. So you see that red is bad and blue is good. So right here you have high parasite rate. So basically the proportion of plasmodium in the body of the kid between 2 to 10 years old. So it's much worse in red, and blue is better. So you see it's correlated with R naught. Places that are red tend to have higher R naught. Places that are blue that tend to have also lower R naught. So we can use this as a precursor to, we can use R naught as a way to measure this scenario. So what we did, what happens now if the global temperature increases by 1.5 degrees Celsius, and we're looking at sub-Saharan Africa, what we see is, because one of the questions in ecology is, will climate change lead to a shift or an expansion of malaria from endemic areas to other new areas? And what we showed that we have this eastward expansion of malaria if there's a 1.5 degrees centigrade increase in global temperature due to warming by 2030, according to this projection. And so we predicted bad news more for Eastern Africa. This is a huge region, lots of big countries. And in fact, if the temperature increases by 4 degrees, we have a much worse scenario for Eastern Africa. You see a lot more red, basically. So more warming means more malaria, but we've shifted more towards Eastern and Southern Africa rather than what the action is right now in Central and Western Africa. Anyway, so I'll skip that. I wanted to do also this genetic epidemiology framework, but because of time, I will probably skip this. What we wanted to do is to have a framework where the So the resistance to insecticide is determined by a single gene with two alleles. Okay, so resistant allele R and sensitive allele S, and we assume random mating, and then we're trying to predict, okay, what is the frequency of the resistant allele and sensitive allele in the population, and so on, and the Hardy-Weinberg condition satisfied, and so on. So we had a model which allows for that, and this is a very complicated model. Also, was 29 equations, but again, we stratify the mosquito population, both aquatic and adult mosquitoes, according to genotype. Okay. Again, we have two types of mosquitoes, so one that's resistant, the other one that's not resistant. Okay, so what is going to happen going forward? Can we eradicate malaria under this kind of situation? Does insecticide resistance cause more malaria cases or not? I mean, that's the question we wanted to address. And we did that. And we actually showed that even though we have resistance, we can effectively manage resistance and also minimize malaria, provided we choose, this is here, we're plotting coverage of IRS and coverage of long-lasting insecticidal. As long as we're choosing this coverage within this white region, we're good. We can actually, within this, so in this parameter space, we can actually both effectively control malaria. This is the prevalence of malaria in humans, and also minimize resistance. So there is an optimal region in parameter space for our best control. We use an indoor residual spraying and long-lasting insecticidal net, within which, if we choose our coverages properly, optimally, we can actually achieve the objective of malaria eradication. And this is when larviciding is not done, so CL is 0. When we increase larviciding, you see the size of the control window reduces. This here, this is the frequency of the resistant allele in female, and this is the frequency of resistant allele in male mosquitoes. And you see that it reduces as you increase more coverage. Anyway, so some possible areas of collaboration. I have to stop now. First, I'm looking for collaboration with people that can help me with data. I collaborate here locally with entomologists that do the experiments in the lab. So lab and field data for fitness, cost of resistance, climate data also at final resolution, data on bed nets, mosquito population ecology data by genotype as well. There's also modeling, coupling immunology, ecology, epidemiology, as Hayriye just discussed a moment ago, climatology, behavior, human behavior, changes in biting behavior, evolution of parasite resistance, assessment of vaccination programs, herd immunity thresholds, and also potential impact of gene drive-based interventions. I actually was planning to talk about that, but I don't have time. CRISPR-Cas will back here and so on. And earlier on today, actually, Professor Glazier and myself were talking about, he was actually telling me about connection between malaria and cancer, so I'm very interested in learning about that as well. And the statistical component, so we need collaboration with data fitting and parameter estimation, uncertainty and sensitivity analysis, and so on. I was going to talk about this. If people are interested, I can discuss that. This sterile insect technology releases mosquitoes into the wild, and they mate with adult female mosquitoes, and the offsprings. The idea is the offspring will not lay eggs that will hatch into larvae and so on. So we have this paper. If people are interested, we can discuss in detail, and we show that it actually works. We have at steady state, we have this mosquito that we genetically modify, actually outcompete the wild mosquitoes, and that's a great thing. But I'm happy to answer questions on this, and thank you so much for your time.
Okay, well, we'll have to have you come back and give another talk. You're always welcome to give more than one talk.
Yeah, sure. Absolutely.
Thank you so much for the two wonderful talks. So the floor is open for questions. Please go ahead to either of our speakers. Richira, thank you for being so patient, and thank you for posting the links.
So I've got a question about your plot of R naught versus the mean temperature
Yes.
in relationship to EIR. It seems like R naught is even more sensitive to the EIP, the intrinsic incubation period, how quickly a mosquito becomes infectious.
Correct.
Where did you get data on that? Been unable to find very little data on how that for Anopheles. For the
that's a great question. So we have the data. So we use the climate data. So our models, the parameters are some of our functions of temperature, some of them are functions of rainfall, water temperature. So we use it. What is it? The climate data, the global climate data. Yeah. So the climate data. So we can have to.
As you know, when a mosquito gets infected with malaria, it's only infectious usually for the last day or two of its life.
Correct. Yeah.
So a small change in the time for it to become infectious can have a huge change, and that's very temperature dependent.
Correct.
Maybe even as much as 1 day per degree C. And so I'm wondering about, did that go into your model?
Yeah. So we had again. So the ecologists do this experiments to collect the data. Okay, they subject the mosquito to different temperature and different level of toxicity, and based on that, we're able to estimate what is the. So what you're saying is, okay, so if the mosquito is infected, so the rate at which the mosquito from the E class goes to the I class, that's
Right.
That's what we actually cover. The ability of the mosquito to survive this sporogonic cycle to become infectious. So that rate is estimated from entomological data actually collected based on the lab experiment that people have done to see that. So different types of mosquitoes, okay, how long does it take to become infectious and so on. So we have data. It's mostly, I think that's the paper from Mordecai, Erin Mordecai's paper, that we have this functional forms for those transitions from exposed to the infectious class. But I don't know if I answered your question.
Okay, we can talk more later. Okay?
Yeah. No, but I like the question. Yeah, because it's definitely true. And that's, again, McDonald's work was, okay, ignore immature mosquitoes, you focus on adult mosquitoes. When are the adult mosquitoes most relevant to us? When can they give us disease? It's only when they survive the sporogonic cycle, when they become infectious. And that's the idea. So that's really, so we do not have to, we can do indoor residual spraying and focus on them when they are most vulnerable. And that's in the stage 2 of the gonotropic cycle after taking blood milk. But if they're infected today, they're not going to be giving malaria to us. They have to wait for that duration of the sporogonic, which is about 9 days, could be longer, depending on the species also.
Yeah, it's that waiting period that's so sensitive to temperature.
Absolutely.
Yeah.
True. Absolutely. The waiting is extremely sensitive to temperature. Even the progression from the exposed class, yes, that is extremely very sensitive to temperature. Absolutely.
Okay, Ruchira, let's go to you next.
Thank you. So I had actually a couple of questions for each of our speakers, but so I'll say my questions first, and then maybe James, you can distribute them. So the questions for our first speaker, one was, so I saw on your slides that you have some viral load measurements that you are, so it's basically about the data that you are fitting to. So did you have data for the antibody levels, and what other data you're fitting your model to? And the second question was about the functional form that you chose for your differential equations, but specifically for the cross-body antibodies, the cross-reactive antibodies, the Y. I saw that you had the positive, in the positive term, it was linear, and in the negative term, it was quadratic. And I was wondering, like, was there some rationale behind this, or was this just what would fit well? So those were my two questions for our first speaker. And then for our second speaker, my two questions were, one was about, you brought up the possibility of eradication. So if there are non-human primate hosts, and there's a vector, so then the parasite can complete its whole life cycle in a reservoir that's completely outside of the human population. So how is there even any possibility of eradication when that reservoir could persist? And the second question was about, you were talking about the effects of climate change, and you talked about the effects of temperature. And I wondered whether you looked at the distribution of water bodies, since the water bodies are essential for one part of the life cycle, and whether the distribution of those water bodies might change in terms of the aquatic part of the life cycle, whether you had taken that into account. So sorry, that was a lot of questions, and then maybe James can manage who
Why don't we start out with the question, maybe repeat the question for our first speaker, and then come back afterwards and ask the second question. They're both wonderful questions.
Yeah, so Dr. Thank you. These are really great questions. Regarding first your first question, it's about finding the data. And I should say that currently my PhD students is focusing on that. And there's a huge challenge because dengue is a human disease that most of the time when we find the viral data, at least, there's always missing data first a few days, because you cannot do experiments with the humans. The purpose to infect them gets blood samples. So it means that my graduate students using different statistical approaches, he's trying to recover some of the missing data, let's say, because usually individuals go to hospital when they show symptoms. In the meantime, there are 3-4 days, there's no way you don't know how the data is like. So I think is really, so another thing is that finding both viral data and IgG cross-reactive and then specific data. So this also, there's issue with that. So I should say that it's a very tough question. Now what we are struggling more with, like, missing data actually. And there's a mismatching data problem, means that although we have primary infection data from some hosts, we don't, and then we have a secondary infection data from the other hosts, we don't know they're the same host. So there's a mismatched data problem. But I think so far, my graduate students, Caleb, is really handling very well. We just need to use some sort of statistical tools to get around this mismatched data and the missing data problem. Regarding your, I will be always happy to talk about it further if you're interested. Regarding your second question, it was about the formulation, the way we formulate the rates for the cross-reactive and specific antibody, IgG antibody immune response. As I mentioned, that for the specific IgG immune response, it's always negative term, it's a saturating form. But regarding cross-reactive IgG immune response, we have a plus saturating term and we have a hill function, which is minus term. So the idea is that we wanted to, so the biological motivation about this formulation that it takes so much, having so much cross-reactive IgG immune response to kill the virus. So what this tells you is that your IgG immune response should be sufficiently large that it should start to kill the virus. It means that when you hit this like saturating functional form of IgG, cross-reactive IgG immune response, and then minus this hill function form, sigmoid function, the sum of these two terms should be negative eventually when cross-reactive IgG immune response large. That means it's killing the virus.
Is there some cooperativity in the
yes, exactly. Yes, we do.
Could you explain what is cooperating with what? Like why is it? Yeah, just elaborate on that a little bit.
Oh, sorry. If you can ask your questions again, like.
So you're saying that there's some cooperativity required for
yes.
the cross-reactive antibody to actually kill the virus, and if there's not cooperativity, then it can enhance the virus. Is that?
Yes. So it's interesting because until a few years ago, at least when we were doing this work, it wasn't clear that it was very big. It was a question why this antibody-dependent viral growth enhancement happening. So I guess the NIV theory, the NIV hypothesis that it is again somehow. So this I'm not a biologist, but let me tell you the way I understood. So it says that to be able to the cross-reactive IgG immune response to neutralize the virus, attach the virus, click with the virus, it takes only so much of them to be around. So if it's not around, then enough, I don't know what is the biological process going there in the meantime, somehow it's allowing the virus to replicate further. I don't want to say something wrong, but I probably would like to talk someone with, but it's just, it's very clear that it's required. I mean, I can understand from this information, it's easy for me to formulate the model, but I really do not know what happens when there's not enough cross-reactive IgG immune response around. I think there's some biological process that probably it would be nice to talk with someone who just actually know these things better than me.
Okay, I got it.
But these are really great questions. Thank you.
Yeah, thank you for the very interesting talk.
Yeah, and also response to your first question, that's also a very nice question. If the mosquito can bite non-human primates, we have this reservoir, which is true. So we had this Plasmodium crossover to humans like 12,000 years ago. Before that, we only have animal malaria, right? We only have malaria, lizards and other, so it's a spillover that happened 12,000 years ago. So but we can still eradicate. I mean, the WHO has a definition of when a country or a region has eliminated malaria. Some 20 countries, including China and Morocco in North Africa, have actually been declared as eliminated malaria. I think they say like 3 consecutive years of no local cases except imported cases, that's considered to be elimination. So it can work. I know there was some study, I don't remember exactly where it was. Some people used this having cattle in the neighborhood where humans are as a strategy for preventing them from getting malaria, so that the mosquitoes will focus more on biting cattle and so. So there are some strategies like that, but I think we can still eradicate. It depends on the definition of what eradication is. It doesn't mean there's zero malaria completely, but that's some standard for. But it's a very good point. If there's this reservoir, just like we're talking with COVID, about COVID, sorry, yeah. It's a reservoir of these bats that are there, like 3,000 different species of bats that can have a coronaviruses. Yeah, we may get rid of this current one, but another one will come because that's circulation. So that's a very good point. It fits in very nicely also with what I've been, or many other people have been talking about, this One Health initiative. To really understand human diseases, we need to know what's going on in animal population, reservoir population. That's extremely important. So it's a great question. I'm glad you mentioned. The second question is about the distribution of water bodies. Yes, of course, if there's no breeding habitat, water provides breeding habitat, that's critical. So monitoring those, that's important. So when I gave the logistic preparation for the egg-laying rate in my model, the carrying capacity is function of rainfall, is current function of precipitation. So that's extremely important. If there is too much rainfall, it washes up the larvae. If there's little of it, malaria mosquitoes need a little bit more rainfall, water than Aedes mosquitoes, for example. Aedes mosquito doesn't need a lot of that. So that's necessary for breeding habitat. I mean, that's definitely important. Yes. But then you may look at places like, say, okay, Vancouver in Canada, for example, it rains all the time, or London, England, it rains all the time, but you do not see a lot of mosquitoes. Okay, so you may have that. It depends also on the soil texture, percolation, and all sort of hydrodynamics that are important at play as well. You may have a lot of these water bodies, but if things are done properly, you may not have, they may not lead to necessarily to good breeding habitats for mosquitoes. But those are great questions. But going back to Mac's question, I think about the last few days of this progonic cycle has been very important. I have also a related response to that is, so we estimated those parameters, temperature-dependent parameters from data from the lab. But there's a big difference sometimes between what happens in the lab to what actually happens in the field. So if the same mosquito is subjected to them. So we're looking for collaboration in that as well to see what the parameterization would have to be, what the functional point would have to be if the data is based on subjecting the mosquito in the actual field, not in the lab situation, in the control lab situation. So I like that.
Okay, other questions?
Thank you.
Hayriye, did you have a second comment?
Oh, me?
No, I think you had your hand raised. Hayriye?
Yeah. So Abba, thank you for the great talk. I have a quick question. I know your models are already too complicated. But I want to say something maybe you can consider.
Sure.
I'm sure you already thought that. So we always talk about the biting rate, but we know we don't talk about how infectious the bites are important. So I read some research that about malaria, I think it's published 3-4 years ago, suggests that actually they look at the inoculum size, how much the virus mosquitoes inject virus to host, and depend on the inoculum size, vector inoculum size, they see that actually in some cases vaccination, malaria vaccination doesn't even work well. They think that when the inoculum size is so large, it's actually vaccine fails because it overwhelms the immune response. This is what I remember. So it suggests that it's not important how much the biting happens, it's important how much each bites are infectious, which is exactly related to within mosquito viral kinetics. So I wonder that, are you ever planning to some way incorporating your work? I know it's already complicated. I don't want to make it more things complicated, but I feel it seems like it's a reasonable thing to consider too.
No, yeah, that's why in the plot we had a great question, Hayriye. That's why in one of the plots for the effect of temperature on R0, we also had a plot on EIR, the entomological inoculation rate. So in the paper that I think was circulated on the link, one of the papers was my postdoc Okune, as the first author. He actually won the Bellman Prize last year. So we had the EIR, actually, the function. So the infection rate, the force of infection is defined in terms of EIR, the entomological effect, which is the number of infectious.
Are you mentioned with the EIR, you are meaning exposed?
Yeah.
Incubation period?
No, EIR means entomological inoculation rate. It is a number of infectious bite per person per unit time.
Sorry, I missed.
Yeah. Yeah, no, not the compartment. No, EIR.
No, sorry, I kind of confused. Because when you talk about the, yeah.
No, but that's okay. But it's similar. Your question is very similar to my question, right? It's all about infectious bites. That's really what's important. If the mosquito is not infectious, even if it bites you 1 million times, it's not going to pass on the plasmodium to you. So it's the infectious bite that's important. That's why we think, in addition to R0, EIR is also a really great metric to use to measure malaria intensity, because it's the ability of the mosquito to survive that progonic cycle stage, okay, to become infectious. Okay, if the mosquito is infected today, it's not going to pass on the disease to you. No, until it survives that period. So the EIR is a measure of that, okay, the entomological inoculation rate, the ability of the mosquitoes to actually transmit the disease per person per unit time. So that's, we had in the paper, actually, we had a definition for it. In fact, the force of infection is EIR times the average duration of infectiousness. That's what it means. The same thing in your dengue model as well. The infection rate, if you have a susceptible human, the ability of that human to get infected from a bite from an infected mosquito is the EIR times how long that mosquito lives, the average lifespan of the mosquito. So that's the force of infection. That's really the vector competence times, the EIR is the vector competence times the ability, times the duration of that infectious mosquito. So yeah, but that's a great question. Yeah, but it's all about the infectiousness of the mosquito. It's true.
Thank you.
Other questions?
I was going to ask, is anybody interested in the biological controls? The use of, because if we have resistance now,
okay, I'll bite on that, because I've always been convinced that Wolbachia would be a more effective way to control malaria than sterile insects. In the sense that if you use the SIT, as soon as you stop releasing them, it will bounce back usually. But with Wolbachia, if you can sustain an infection, you can totally block transmission. So, yeah, so talk about biological control. What's your thought on that, Abba?
No, actually, the reason I mentioned that because I know you're here, because you do great work on Wolbachia. So that's, but yeah, my thought, I think it depends on which one. So SIT, is the idea is to release male mosquitoes that are sterilized, okay, that through this irradiation process, so that when the mating happens between this modified mosquito, sterile male mosquito was a wild female mosquito, the female mosquito will conceive, will lay eggs, but the eggs will never become larvae, will never become. So it's a way of altering the sex ratio. Yes, so the question is, when you have this interventions, this biological control, that key questions, okay, well, how many do you need to release? Okay, so logistically, how many how frequent, how often do you have to do this? So Google tried this, I believe, in Fresno County during 2014, I think during Zika outbreak. They released like 1 million mosquitoes. Also, you have to make sure you release at the right location with maximum density of adult female mosquitoes. So it didn't quite work for Google, but yeah, I agree with you. So what we showed in the paper that we published is if you release like 100,000 mosquitoes every 2 weeks for a period of 2 years, you're able to bring down the mosquito population significantly. But yeah, if you stop it, then yes, it's possible for a rebound, but you have to then combine this with other interventions. You have to combine it with, let's say, continue to do IRS and long-lasting insects. With Wolbachia, yes, this is naturally occurring. It's a mosquito is infected with Wolbachia, fantastic, it's already there. But you have to make sure also that these mosquitoes are always there, infected mosquitoes are always there. So are there mechanisms to produce them? But there's another one, CRISPR-Cas system. That one is actually really fantastic. So for example, you modify the mosquito to make them not being able to have a plasmodium, and then you release them into the wild. And I think studies have shown, I think Mark Lipsich and a few others, either Mark Lipsich or Martin Novak, I'm confused, one of them did this work, probably Novak, that within a few generations, the wild mosquitoes is gone. You only have those modified mosquitoes surviving. And that's permanent, basically. It's not, unfortunately, sometimes it's not reversible. You cannot reverse that. Once you modify the mosquito or release them, it cannot. So that's one issue, but that can be done. But yeah, I agree with respect to sterile insect technology. That has been used in the past in the United States in the 1940s, like pest control against other pests, and it was very successful. With respect to mosquitoes, mosquito-borne diseases, yeah, the jury is still out, but at least we showed, many others have showed also that it can work, but you have to also combine it with other stuff. But I like the great work you've been doing all back here. That's fantastic work.
Maybe you'll come and give us a talk on that.
Absolutely, yeah. Anytime.
Well, the big thing that, one of the big things we learn is how important it is to include spatial heterogeneity. Because these ODE models that we've been talking about assume that the density of mosquitoes is the same everywhere. And when that's really not the case, especially when you release genetically modified mosquitoes or Wolbachia infected mosquitoes, it may be very high infection in one place, but low elsewhere. And that has an incredible impact on the dynamics. So.
No, yeah, there are various levels of endemicity for malaria. I mean, holo-endemic, meso-endemic, and all that. Yeah, certain places, even within sub-Saharan Africa, you see, like I was saying before, in the East.
Actually, James, someone who I think you would enjoy here is, Hayriye also mentioned it, is Jolyn Chu. She's at UT San Antonio, and she's been working with me. She's been doing the spatial modeling.
Oh, nice.
Of it, and gives an excellent talk. So I think if this group would like to hear a little more about these techniques, I think Jolyn would be an ideal speaker.
Excellent.
Yeah.
Well, as somebody who does spatial modeling, I always enjoy hearing spatial modeling.
It's just in its infancy now, but I think it's crucial because things that look great with the ODEs, you saw that backward bifurcation diagram.
Yeah.
Yeah, you may be above the threshold locally, but 100 yards away, you're below it. And
correct.
So the question is, you can get these traveling waves, do they spread out or do you get self-collapse? And it's the dynamics is a little closer to the real world and a lot more interesting. So.
No, but that's great. Also, I think there was a study also with SIT in Australia that shows, well, you may, within the boundary where you're releasing the SIT mosquito and doing so well, but then you have migration from local areas onto
right.
So how do you?
It all works until a big wind blows through.
Exactly. And that's the problem Google had, I think, when they released their mosquitoes from the drones in the terrestrial country. The wind just took away the mosquito. I mean, so there are all these logistical issues. But I'm also very interested, Professor Glazier mentioned about the connection between malaria and cancer. I did a little bit of such and see that it's Burkitt lymphoma that is associated with malaria, and it's correlated. This BL is correlated, geographically correlated with malaria. So the more malaria, the more of that. And you see that's a kind of co-infection with another virus, is EBB, which is I can't remember the name, but it's some kind of herpes virus that is connected to malaria. So the link between malaria and cancer, I didn't know about that until today. I think this is very interesting. If there are people who are interested in collaboration, I would be happy to talk.
Lorenzo, you've been very patient. Go ahead.
Hi, thanks for both the talks. They were both very exciting. I got very interested about this latest aspect of spatial heterogeneity. Well, I'm interested in other aspects of spatial heterogeneity, but this rings a bell. This is very naive, probably, because I don't have really a good background in malaria. But is this heterogeneity regulated? So there is some function that says, all right, or some parameter that makes mosquitoes be more dense in one place than another. And the follow-up question would be, if we know that there is some element that regulate that, could we exploit it and put some attractor in the desert, or well, they won't go in the desert, somewhere that there is not so much population, etc. Might be that strategy.
Yeah, so that's a great question. Yes, it depends on the availability of breeding habitats, right? So that's where mosquitoes go, depends on the level of, let's say, construction work that's been done, for example, irrigation, all of those things. Like here in the state of Arizona, in the Gilbert area, close to where I am, there's a lot of construction going on. You see that they have more per capita, more mosquitoes in that county, in that region, on that location compared to many other locations within Phoenix area, metropolitan area, for example. So it depends on what we as humans are doing to create the breeding habitat, or people are not taking care of their pools, for example, in swimming pools. I mean, are they doing irrigation, or they're clearly is always connected, is anthropogenic, is always connected to what we do. So the spatial heterogeneity is related strongly on what we assume, and it's similar with dengue mosquitoes. So here in Gilbert area, we're talking about dengue mosquito. Oh, hi, great to see the baby. Great to see.
Yeah.
She's waiting for her turn to ask you a question.
Yeah, please ask the question. Hello.
Yeah, she's the youngest member of this group.
Congratulations, it's great.
Thank
Hello.
Welcome, welcome.
She'll be talking to us about
very
backward
interested.
Yeah, she's very interesting, your talk, Abba. I can
Very
say.
good. I can
She
see that,
doesn't
yeah.
say that nicely.
No, it's great to see
Yeah,
her.
I actually had the question too about, sorry, I don't want to interrupt you, but
No, go
one
ahead,
of
go
the
ahead.
very interesting things about the backward bifurcation is that, you know, we have two local attractors, right? Mathematically
Yeah.
speaking. One
Yeah.
of them is like infection-free attractor, and one of them is coexisting infection endemic equilibrium. One cool thing is that if your initial conditions in the basin of attraction of endemic equilibrium, solution eventually converge there. If it's in the basin of attraction of the disease-free equilibrium, it converge. So idea is that if your initial condition in the basin of attraction of endemic equilibrium, what sort of temporary control strategies you can implement that you can push the solution from basin of attraction of endemic equilibrium to the disease-free equilibrium, basin of attraction of disease-free, which means that disease can eradicate. Yeah, I find it very like interesting. I apply this context to avian influenza. I think I find it in terms of epidemiology, in the context of epidemiology, this is one of the most important things about backward bifurcation, actually.
No, I agree totally. That's a great question, actually. Yes. So I don't know if I need to show the picture, but those of you have seen the, if you want, I can share again. But in the backward bifurcation region, we have two attractors, as she mentioned. You know, the stable disease-free equilibrium, which is what we want. We want a scenario whereby at steady state, as we run the simulation long enough, there's nobody infected in town. Okay, nobody has malaria. Basically, that's what we mean. Or by the WHO standard, like okay, less than I don't know, 3,000 imported cases or whatever, or three in a country. So, but then the other one is, even though the reproduction number is less than one, you have the other stable branch, which is endemic, which is positive equilibrium, which means there are more people, there are people infected in the population. So how do you bring back the dynamics down to the disease-free equilibrium when you're in that region where you have biostability? Because in the biostability situation, which one you converge to depends on the initial conditions, depends on how many people are infected. So what I would say there, because for me, the way I would think of it is you'd also have maybe intensify more treatment, because treatment, that's the way to reduce the number of people who actually have the disease, right? So you treat people, they recover. And with malaria, the more times you get it, the better the immune system is. So that's immunity build up as you get more infected. But artemisinin this is the best we have, is combination therapy. So the more of that you use, the more you reduce the number of infectious or infected humans. And then you can bring down, even though R0 is like 0.8, you are in the backward bifurcation region, you can bring it down to the basin of attraction, which is a region containing that attractor, okay, within which all the initial conditions will converge to that attractor. So we can.
Sorry,
go ahead.
Another thing actually is about how much you need to reduce the infectious reservoir to accomplish, you know, like pushing the solutions to the infection equilibrium. This is another question, right?
Well, yeah, but that's a great mathematical question. That's an open question, I think. To determine even the exact location of the saddle point that separates the basin of attraction of the disease-free, Cameron is, where's Cameron? Cameron should be answering this question. So he does a lot of.
He's teaching right now. This is why
he's teaching. Okay, you can ask him. Yeah, he should answer that, because the exact location of the saddle point, which separates the basin of attraction of the endemic and the stable one, is not to determine that. Theoretically, that's not an easy question, even numerically. I mean, so yeah, that's how much do you have to do to bring it to, so basically what you're asking is how much do we need to do to come below that separatrix which separate the basin, and that's not a trivial question. But all we can say to entomologists is we just need to do a lot more in terms of control, even though, so what it really means, backward bifurcation means, is that we have to do a lot more. You know, just bringing R0 to less than 1 is not enough. We have to bring it much, much lower than 1 outside the backward bifurcation region to have a realistic chance of eliminating the disease. It's the same thing with COVID. If you have vaccination, which is imperfect, we inherently have a backward bifurcation. So even though right now we did the simulation yesterday or 2 days ago with Omicron, we had R0 of 0.96, and Delta has R0 of 0.28. So we think that Delta is going or is gone, but Omicron is close to 1. We have to keep on doing it. It's not enough just to say, okay, we no longer masking. No, it's going to rebound. It's going to come back again. So we have to, because of backward application also. So bringing it to 0.96 is not the same as bringing it down to 0.2 or 0.3. We have to do a lot. So backward verification makes the effort to control a lot more difficult. It makes the requirement on interventions to be a lot more stringent. We have to do a lot more. We have to keep on using indoor residual spraying. We have to keep on using bed nets. We have to hopefully come up with a vaccine that will be a game-changing moment like it was for COVID. Yeah, it just makes it more difficult to control disease if you have a backward verification.
I mean, given that malaria is an extremely complex vector-borne disease, I don't know, things get harder because the way malaria functions, I guess how the vector component functions and how the parasite discretely produce and come, which is like all makes it like such a complex problem.
Absolutely.
Like
it's a complex
scale. Yeah.
Well, that's the reason we've not been able to get an excellent vaccine against malaria. I mean, that's the reason, it's the complexity. But then that's great for grad students here, for postdocs. I mean, to take this as a challenge because you have multi-vector, multi-host, multi-parasite. I mean, a lot of mathematical challenge, even data fitting the model with data, that itself is not easy. It's not trivial. You know, it's a serious statistical question, computational question. Come up with a computational method for solving the problem. You know, it's not easy. But doing the analysis, but that's why we have grad students. So I had a PhD student who got the Bellman Prize. He was the one who did the analysis for the barcode verification and other stuff. We
always give the hardest problem to our graduate students when it comes to data.
Of course. Well, they do all the hard work. All we do is we talk about it. I mean, but they get all the credit. They should get all the credit. Yeah.
Well, officially, we're out of time. So I want to thank our speakers for 2 wonderful talks and a very, very stimulating discussion. And I hope you'll both come back and talk about additional work. I had a question I wanted to ask about the paradoxes of the vaccination strategy when you have a population optimum that's not optimum for individuals. But I'll have to save that question for the next time you come to speak to us. And please do send me the suggested people we should have speak, because we would love to continue this conversation.
For what you just mentioned, I would suggest you invite Chris Bauer from University of Waterloo. Game theoretic question on the right of an individual against that of society. So it's a game theoretic stuff. So let me just write his name in the chat. Chris Bauer, Waterloo. I think he'll be an excellent speaker for this.
Yeah, Dr. Glazier, thank you for the invitation. I will send you some suggestions on that, including my grad students. I think he can give. I was wondering, is there any place that bringing some interesting problems? I think some of the question Dr. Datta mentioned actually, which my student trying to attack, we will create, if there's a discussion platform that we can actually talk about these problems, maybe there's a place to collaborate together.
Mm-hmm. Well, I mean, you certainly are welcome to come and present an open problems talk. People have done this. Some people, this forum is open. And if you have a suggestion for creating a discussion group, we have a variety of subgroups, but they take work to organize. But if you have a set of open questions you'd like to present to the community, you're welcome to do that.
I will say open and ongoing, actually. Yeah.
And so
I think a discussion on spatial heterogeneity in mosquito-borne diseases in general, I think that will also be extremely interesting, as raised by Mac and also Lorenzo and a few others. So I'll be very interested if there's something like that. I'll be interested to come and discuss that.
Wonderful. Well, we look forward to having you all back. And Mac, we do need to get you on the agenda for a talk. Lorenzo, I hope you're taking notes about the invites. Okay, well, thank you everybody again. I really appreciate it. And I will see people next week.
Thank you, James.
And thank you for the invitation and for the comments. Very nice. Thank you. I appreciate it.
Goodbye.